Swayam Prabha. Digital India. Educated India.
Good day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. We have now learned about different data formats, we have learned about quality control and we have done the pre-processing steps and we have done all of those hands on. So, you have seen how we can go through all these steps. So, we are now ready to move into the downstream analysis or the processing steps, main processing steps, and as part of that we are going to discuss read mapping today. Okay. So, these are the concepts that we will cover in this class. So, what are the challenges in read mapping? We will talk about the brute force approach and we will talk about this evolution of this mapping algorithms. So, we will talk about BLAST and BLAT and why we need better and more efficient algorithms. So, these are the keywords that will come across throughout this presentation: mapping, seeds, query and reference. So, again going back to the flow chart, right, of NGS data analysis. Once you have this quality control done, you can do this pre-processing if it is required, and then you move into the mapping or assembly depending on the problem that you are working on, right. So, if your goal is to do signal nuclear polymorphism analysis or transcriptome analysis, you will go into the read mapping side. So, you will have a reference genome against which you will map your sequenced reads. If you are working with a with an organism where there is no reference genome, you will do the assembly, right. So, today's focus will be this read mapping, right, where we will discuss certain challenges etcetera and the approach, and read assembly we will discuss in the later part of this course. Okay, so, the mapping problem, just to remind you, right. So, you have this stack of reads on the left, you can see, and the goal is to identify where these reads come from in the reference genome, right. So, whether these reads come from chromosome 1, chromosome 5, chromosome 10 etcetera, that is the goal of this mapping, right. So, for example, if we look at this schematic or this animation, right, we have this read that is kind of we found a match here in this position, here in this reference sequence, right, and this is the mapping that is done. We can continue, right, with these reads and we see, okay, this read matches there and it is mapped there, okay, and we continue this process, right, for all the reads. Finally, we get this kind of picture where we have reads, all reads mapped against the reference genome, we know their location. Now, this sounds very simple, but there are of course many challenges which we will discuss today, right. So, what are the major challenges here? Okay. So, the first challenge is that we work with large genomes. Okay. Most of the data that you will see for example, for human genome, right, this will be huge. Okay. So, this is like 3 into 10 to the power 9 bases. Okay. So, if you work with plants, mammalian genomes, these are really big genomes. Okay. So, what is the problem with big genomes? Why is that a challenge? Because then you have many potential locations where this read can come from, right. So, you have to check all these locations, right, and this takes time and computational resource. Okay. So, this is why the large genomes are a challenge for us. Then you have huge number of reads. So, when we were discussing this NGS technologies, right, we looked at the throughput, right, the number of bases that we get from each experiment in those platforms, right, and these reads are in millions or billions, right. So, you can in today's experiments you can get billions of reads quite easily. So, if you can visualize in your mind that you have to repeat this process of mapping, okay, for each of them, okay, if you have 1 billion for each of them, you have to repeat this process, you have to find the position in that big genome. Okay. So, you have this at one hand you have these big genomes, right, meaning many potential locations, and at the other hand you have huge number of reads. Okay. So, if you combine them together, the problem is the time requirement. Okay. So, you will probably need a lot of time to actually complete this process. Okay. So, if you had only let us say few reads that you have to map against human genome, then it is fine, you can do it maybe in an hour or 2 hours, etc. But here we are talking about large number of reads, and that actually compounds this problem. Okay. All right. So, let us look into other challenges. So, we are dealing with mostly short reads. If you kind of are working with kind of short read data, because you want to do single nucleotide polymorphism analysis, right, you want more accurate data. You can also have a lot of mutations and sequencing errors in the data, right. So, this happens whether you are working with short reads or long reads, this sequencing error will be there. What does it mean? If you have mutation or sequencing error, means you will not find perfect match, right. So, you can imagine this mapping problem as a string matching, right. So, you have this read which is kind of a combination of letters, right, ATGC, and at the other hand you have the reference sequence which is also a combination of letters ATGC, right. So, it is at the end its mapping is kind of string matching, right, where there is perfect match in the genome. So, here if you have a sequencing error, one of these bases will be changed to something else, and what you will see, there is no match, you probably will not find any match in the reference genome, right. So, there is no perfect match. So, this is something you would have to consider while you are doing the mapping, right. So, any algorithm, any tool that does mapping will have to consider that there may not be perfect match, and we would have to find the best match. Okay. So, this is again a challenge, right. So, just to kind of represent this schematically or in an animation, here you have like, so if you do not have this mutation in red, this is a perfect match, right. We find say okay, there is a perfect match and this read comes from this position. But if you have this mutation in red, then you do not have perfect match, and it makes this mapping problem more challenging, right. Then you have one more challenge, which is the repetitive regions in the genome. Okay. So, human genome or other mammalian genomes, they contain a lot of repeat sequences, right. So, one sequence repeated multiple times across the genome. So, any read that comes from these regions, right, it will have multiple matches in the reference sequence. Okay. So, here it is, right. If you can put this in a schematic figure, right, here is the read, and this comes from repetitive region that is present in two positions of the reference chromosome. Now, when you are doing this mapping, how do you decide, or can you even decide, right, whether this read comes from this location or this location, right. So, this is again that creates ambiguity here. And finally, there is another challenge. This is a specific challenge for RNA-seq data, right. So, we will talk about this in a bit more detail when we talk about transcriptomic data analysis and read mapping in RNA-seq data. So, what happens in RNA, right, when you are processing RNA samples, right, and this mRNA you have splicing, right. So, when you have these splicing events, you have these exons, right, that join together and they cut out the intron in between. Okay. So, when you generate reads, it will just give you the exon sequences, it will not give you the intron sequences, right, because this sequencing is done on RNA, right, from RNA to cDNA and then on then that is sequenced. So, you will get these exon sequences only, not intron sequences. Now, when you go and try to map, right, what it means is that you have to do something called gapped alignment. Okay. These two parts of the same read will match to two different locations in the reference genome, right. So, the aligner, the program that you are using or the algorithm that you are using needs to count for this, right, needs to know that this can happen, because we are dealing with RNA-seq data. So, these splicing events can happen. So, we should be aware that there could be such mappings where you will have these two different locations for one read. Okay. So, again there are different ways to address these challenges. We will talk about this, especially this splicing in RNA-seq data when you go into transcriptomic data analysis. All right. So, we have kind of understood the challenges, right. We are working with big genomes, we have huge number of data, then on top of that we are not working with ideal data. So, we have sequencing errors, we have repeat sequences, etc. So, to make our life more difficult. Okay. Now, coming to the simple question is this is regarding the search process, right. So, this is the major component, okay, of the algorithms. Okay. How do you find the reach sequence location in the reference genome, right, and what is the search strategy. Okay. So, this is what we will start with today, and we will talk about different approaches and see how these approaches work and what would be the limitations of each of these approaches. Okay. So, you can start with something called a brute force approach or a trivial mapping algorithm, right. So, what does this mean is that we can simply say, okay, we will have the reach sequence, we have the reference sequence, and we can slide that reach sequence along the reference genome sequence and see if there is certain matches around, right. So, once we slide, we count number of mismatches compared to the reference sequence, right, and if the number of mismatches is below a threshold, right, we can say, okay, this is an alignment, this is where the read maps. Okay. So, this counts for the sequencing errors or mutations that might be present in the data, and there might not be any perfect match, right. So, let us take some examples and then we will understand how this works and the limitations of each of these processes, right. So, here is the reference sequence, for example, right, this is our genome data, for example, and we have the read, which is the query sequence, right, and what we want to do is we want to find this query sequence in the reference genome, and we want to find the location of this read in the reference sequence. Okay. So, based on this brute force approach, what we do is we put this read, right, against this reference sequence. Here we start from the first position, right, this is the reference on top and then query here, and we count number of mismatches. Okay. So, number of mismatches here is about 13 and we put the threshold as 3, right. So, any kind of position where you have less than 3 mismatches, we will say, okay, this is an good match, this is an alignment, and the read maps to this position. Okay. So, we can set these thresholds at different levels depending on what is the expected error rate in our sequencing data, etc. Okay. So, this is clearly like you have more mismatches than the threshold. So, we cannot say this is an alignment, right. We move along, right, we are sliding along the reference sequence and we now see again, we again count the number of mismatches, here it is 12, again it is higher than the threshold. So, we move, we continue, right, here there is slight decrease 8 mismatches again, but this is again higher than the threshold that we have set, and we continue this process until we reach a point where you see, okay, we have 2 mismatches and it is below the threshold which we have set. So, this is the location of this read. Okay. Now, as you can see, this is a very tedious process, right, especially for big genomes, right, where you have like huge number of positions that you have to search through. Okay. And how do you also count these mismatches? You also you have some algorithms that will actually count these mismatches. Again, these are also very slow, right. So, we can look at the drawbacks, right. So, one is the time requirement. Okay. The sliding window approach along with this mismatch counting, right, both contribute to the time required. Okay. So, let us take this example, right, let us say genome size is about 3 billion bases, right, like human genome. We have number of reads which is about 30 million reads that we have generated. This actually is on the lower side because many times you will get even more, right, billions of reads from the sequencer, and let us consider the read length is about 150 bases. Okay. So, now you can imagine the scale of the problem, right. So, you have to check this 150 base read along this 3 billion base long genome. Okay. So, you will have about 3 billion comparisons approximately, right, slightly less, but it is approximately 3 billion. Now, let us assume, right, this time for each comparison is about 1 nanosecond. Okay. So, about 10 to the power minus 9 second, right. This is again quite optimistic. This will again will depend on the computer that you are using, the system that you are using, right. So, let us be optimistic and say, okay, this is quite fast process, this mismatch counting, and so for each comparison you have this 10 to the power minus 9 seconds. So, how much time you require if each comparison takes about 10 to the power minus 9 seconds, it is about 3, you have 3 billion comparisons, right, into 10 to the power minus 9, and you have to do this for 30 million reads, right. So, we multiply with number of reads here which is 30 million. So, it comes about 9 into 10 power 7 seconds, which is about 1000 days, right. So, approximately 3 years it will take to complete this mapping if for 30 million reads, right. So, you can do the sequencing experiments within a week or 2 weeks, and then the mapping takes 3 years, right. That is not a good process, right. So, this means this will not work, right. This is something we cannot utilize for mapping NGS data. Okay. In addition, what you have is that you may not always find the best match, right. So, as we said, like we have this threshold. The moment we have a match where the number of mismatches is below that threshold, right, it will stop, the algorithm will stop. Okay. And this means it might not always find the best match, right. There might be some match somewhere else which is even better, right. So, that if we stop there, it will actually kind of miss the best alignment, right. So, this is one of the threshold. Now, another thing might happen is that if you want to find repetitive sequences, right, again if you stop after this, if you reach below the threshold, right, you will not find these repeat sequences in the genome, right. So, multiple mappings, but if you want to find these multiple mappings, you have to scan all the positions, right. Again this will probably take more time. Now, you probably understand the drawback. So, we cannot utilize this brute force approach, right, because it will take too long. So, the question is, are there alternatives, right. So, can we do better in certain ways, right. So, as we have seen, trivial mapping is very inefficient, right, because most of the comparisons will give you large number of mismatches. So, these are like unsuccessful attempts, right. So, we are making a lot of unsuccessful attempts and we have just 1 or 2 successful attempts, right. So, only few locations will show some match. Okay. Now, so what we can do is maybe we can some way we can reduce this number of comparisons, this unsuccessful process, right. So, that will reduce the time. Okay. So, again if you want to compare the whole read sequence for match or mismatch, this is also time consuming and it also requires memory, right, because we utilize something called dynamic programming for mismatch checking, right. So, if we are using this dynamic programming using Needleman-Wunsch-Smith-Waterman algorithm, again this takes a lot of time, right, if you are doing this for the whole sequence, right. So, these are the limitations of this brute force approach, right. So, there might be some other alternatives that can do better perhaps, right. And one alternative that comes to our mind is the BLAST, right. So, we probably have used this BLAST for searching some sequences against databases, right. So, it seems quite fast, right. If you are searching one sequence, you can get this data within few seconds or so, right. You have seen this yourself, right. So, let us look at how BLAST works and whether we can apply BLAST to our problem, right, to mapping problem. Okay. So, BLAST works by something called seed and extend, right, and it does something called local alignment. Okay. Okay. So, what are the steps in BLAST, right. So, you have to generate seeds from the query sequence, which is the read here, right. So, if you run BLAST, you will see this query sequence and subject sequence. So, here the query sequence is the read and the subject sequence is the reference genome. We can specify, okay, this is the reference genome against which we want to do this BLAST. Okay. So, first you generate seeds from the query sequence. So, we will talk about what these seeds are. These are actually substrings, only a small part of the full sequence of the read. Okay. Then we generate neighboring sequences of the seeds for a given match threshold. Okay. So, we say, okay, we also kind of account for sequencing errors or mutations, and we kind of look at these neighboring sequences or neighborhood sequences. So, what we will take an example to understand this better. Then we search these seeds against the reference sequence and find seed heats. Okay. So, and find the locations where these seeds actually heat, right, and then extend these seed heats until we reach an alignment score. Again, we can set a threshold for alignment score, right, and once we reach that threshold, we cross that threshold, we extend those seed heats. Okay. So, we will take an example to actually understand this process better. Okay. Again, we come back to the same example, we have the reference and the query. So, here the query is the read sequence. Okay. So, the first step is break down this query into seeds. So, we can choose different seed lengths, which we say k, right, which we denote by k here, and we can take k equals to 5 for example in this example here, but you can choose a different case. Okay. So, we are breaking down this query into seeds of length 5, right. How do you do this? So, you start with this seed, right, first 5 will give you the first seed, then you will have the next 5, then so on, right. So, you can see like we can write down this, you kind of have a sliding window and take 5 base at a time, okay, and that gives you the seeds. Okay. So, for each seed now we need to generate these neighborhood sequences. Okay. So, we take one seed, right, and what are these neighborhood sequences, right. So, neighborhood sequences of the seeds with 4 matches, right. Again we can specify whether we want 4 matches or 3 matches, right, depending on the sequencing error that we want to accommodate in our analysis. Okay. So, for seed CAGA, right, so we look at the neighboring sequences with 4 matches, means 4 bases will be identical to the seed and only one base can vary, right. So, here are all the neighbors of this seed. Okay. Now, what it means now is that we are accounting for this sequencing error at different locations of the seed. Okay. So, in the next step we then search these seeds and the neighbor sequences in the reference sequence, right, and since we are considering all the variations, there should be a perfect match. Okay. And again if you kind of sliding across this reference sequence, you will see at this position you have this match. Now, you might ask like how is it different from the earlier brute force approach, right. So, here we are taking actually a small part of the sequence, right, and this actually finds this actually can check for match only, right. We are not looking for mismatches. So, we do not need to use something like dynamic programming or more time consuming algorithms, right, because we are looking for match, perfect match, you can simply kind of say look at identity, right. And so, that process is much faster, but we still have this sliding thing, right. We are kind of looking at linearly across this reference sequence. Okay. So, once we find this match, okay, we kind of extend in both directions, okay, and we extend until we cross this alignment score S, right. So, if we cross this threshold score S, then we say okay, we have found some match, right, we have mapped a read. And then we stop, right. When do we stop? After we have crossed the S, so we stop if we see like okay, the alignment score is falling below a threshold T. Okay. So, we can define this S and T depending on our requirements. Alright. So, this is the whole thing, right. So, the idea is we break down the reference sequence instead of matching the full query sequence into the reference, right. We are taking a part of this query sequence and mapping against the reference, right. So, this kind of speeds up the matching or mismatching process. Now, the drawbacks, right. So, as I said like there might be some improvement in this mismatch step, right. So, again going back to the same example, we are looking at the same data, right. We have 3 billion bases in the reference sequence, we have number of reads 30 million and we have similar number of comparisons, but let us assume that time for each comparison now is 10 to the minus 10 seconds, right. So, we have improved there, right. So, we are doing that faster. So, what it will mean is that the total time that will be required, right, that will be actually less, but still it is quite high, right. It is about 100 days. Okay. So, it is still not acceptable, right. It takes about 3-4 months, that is not acceptable for us. Can we do better? Is there any way we can improve on this, right. So, the question is how, right, how do you actually improve on this? Now, one of the things you probably have noticed now is that whether we are going with brute force approach or with BLAST, we are kind of linearly searching through the reference sequence, right. We are kind of sliding through the reference sequence, right. Instead of that, can we also seed or index the reference sequence? Can we create some of the can we fragment the reference sequence, right. We can take out parts of the reference sequence, right, and then search against the query, right. So, why is it better? Why is it a better idea than what we have done so far? It is because we the query sequence is smaller than the reference sequence, right. So, if you are searching linearly through the reference sequence, it takes more time to search a single seed or single sequence, right. But if we do the opposite, if we create those seeds from the reference sequence and search against the read sequence, this will be faster because the read sequence is smaller, right, in length. Okay. So, we may have to search more seeds, but the process of comparison for each seed is much faster, right. So, this is kind of the idea, and there is a tool that kind of does this. It's called blast like alignment tool or BLAT. Okay. So, how does it work? It seeds the reference sequence. Okay. So, these are the steps. So, it seeds the reference sequence and then it searches through the query sequence. Okay. So, again we go back to the example, right, where we have this reference sequence and we have the read sequence or the query sequence, and we take the same seed size and we generate seeds of length 5 from reference. Okay. So, again we follow the same process, right. We start from the beginning, we take seed of length 5, right. We count only 5 bases, that is a seed, then we move on, right, to the next base, take next 5 and move on, right, so on, and these are the seeds that we will get, right, as we move along, right. You can do this for the whole reference sequence. Okay. Now, once you have found this, right, you can also generate neighborhood sequence of the seeds, okay, considering all the variations, and then you can search through the query sequence. Okay. Now, it's the reverse, right, compared to what we have discussed now before, right, BLAST. So, the whole process has reverse. Okay. So, without going into all the details, because we have seen how you generate these neighborhood sequences, you generate those for the seeds here, and then we search through the query sequence is something like this, and then you find heaps. Okay. Again, you expect perfect matches because we have considered these variations, right. When you are considering these neighborhood sequences with certain matches, we are considering for sequencing errors, right. We have accounted for sequencing errors. So, you should find perfect matches, okay, and you do not have to apply dynamic programming or something else to actually find number of mismatches, etc. Right. So, once you find this heat, you just extend again till a threshold alignment score S. Okay. So, this is something that you might want to see, right, whether this actually works and whether we actually improve on time. We can actually again look at the time calculation, right, and maybe we can say we have improved upon this a lot more, right. So, now every comparison takes even less time, 10 to the minus 11 seconds, right, and you can calculate the total time, it is still about 10 days, right. So, this is something again that is probably not good enough. Okay. For 30 million reads, if it takes 10 days, if you increase the number of reads, if you are processing billions of reads, it will take even longer, right. So, if you are processing 300 billion reads, it will take 100 days, right. So, this will increase the time, and that is still not acceptable, right. We want to do this within few hours, right, maybe a day or two, right, not 10 days for 30 million. Okay. So, what it means, we need more efficient algorithms. So, whatever we have discussed, the brute force approach, BLAST, BLAT, they are not good enough for this problem. Okay. And this is because for number of reads that we get from the data is often more than 30 million, right, in billions, and will require a lot more time. Okay. And one of the problems here with all these algorithms is that majority of comparisons are not successful comparisons, right. So, we are doing these comparisons and most of them will not give any positive result, right. So, we are spending a lot of time in doing these unsuccessful comparisons. So, can we reduce the number of such comparisons, right. So, if we can reduce this number of comparisons, then we can improve on time, right. So, we can probably focus on the successful comparisons and we can get to the results much quicker. Okay. So, these are the references that we have talked about today, and to summarize, we have seen that if you want to map a large number of reads, this is quite a challenging problem, right, that requires a lot of time and probably computational resource. So, this is something we would have to think about carefully, right. There are two important considerations, one is time, right. You do not want algorithms that take too much time, because that is not what you want, because you will be done with sequencing within few days or a week, and if your program takes few weeks, that is not good enough. Okay. And also you do not want to use too much memory, right. This is again very important, because if you can run this analysis in your desktop system, that will be great, but if you cannot, right, if it takes a lot of memory, if you need sophisticated servers, large computation resources, okay, that will again limit the accessibility. Alright, so these are two important considerations. What we have also seen is that BLAST is too slow, right. So, we cannot of course use brute force approach at all, that will probably take forever, but even BLAST that people used for searching through databases, this is also too slow for mapping. When you have this large number of reads, this number approaches billions, this process takes too long, right. We have seen an improvement over BLAST, which is the BLAT, right. It takes less time than BLAST, but it is still inefficient if you are going with billions of reads, okay. And this means we need more efficient mapping algorithms, okay. And this is something that has developed with advancement of sequencing technology. As people have seen, we are getting more throughput from the sequencers. There was a need for more efficient mapping algorithms, and that is why we will see, like in the next class, there will be a plethora of algorithms that can do this job in a much more efficient manner. So, we will discuss these algorithms over the next few classes, and we will then look into the best one, right. We will do a comparison, we will look into the best algorithm, and then we will also discuss the tools that can do this very efficient. Thank you.